Buenas tardes, compañeros. Bienvenidos al Seminario Departamental del Centro de Tecnología Genómica. En esta ocasión tengo el gusto de presentarles al maestro en Ciencias Luis Antonio Castillo Alejos como el nuevo jefe del Departamento de Posgrado, quien nos estará apoyando en estos procesos, en este seminario, y estará para atender sus solicitudes en el área del control escolar y todas las constancias que requieran los alumnos. Entonces, le voy a ceder la palabra al maestro Luis Antonio para que continúe con el seminario departamental. Adelante.
Bueno, buenas tardes. Gracias, doctor, por la presentación y buen día a todos. Sean bienvenidos al Seminario Departamental, que en esta ocasión está a cargo del Laboratorio de Interacción, Ambiente, Microorganismo, y la ponencia del día de hoy nos será compartida por el maestro TUNDE, OLARINDE, OLANIYAN, con el tema Resistencia microbiana, caracterización y análisis molecular de cepas del grupo ESKAPE de la subregión hidrológica del río Soto, Marina, Tamaulipas. Maestro, adelante. Demos la bienvenida al maestro OLARINDE, por favor.
Good afternoon, everybody. My name is OLARINDE, and I'm here to present my progress report on antimicrobial resistance, characterization, and molecular analysis of ESKAPE group strain from Soto, Marina. First, the word ESKAPE is an acronym for 6 nocosomal pathogens that have been known for their multi-drug resistance and virulence nature. They include 2 gram-positive bacteria, Enterococcus faecium and Staphylococcus aureus, that have been known to cause a lot of infections in clinical settings. Also, we have 4 gram-negative bacteria, which include Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and lastly, Enterobacter species. These organisms have been known to also cause pathogenic infections, also in clinical settings. This incidence or incidence that has to do with the resistance with ESKAPE group strains is as a result of, because they have diverse resistant mechanisms. One of such as the antimicrobial inactivation, either by beta-lactam modification or by aminoglycosides acid modification. Also, it can be by bacteria target site modification. As you can see, it can be by cell wall alteration or by cysteine sRNA methylation. And in some cases, the bacteria can make the lipopolysaccharide membrane look gram-positive, so that they are resistant to, especially in gram-negative bacteria, so that they are resistant to colistin. Also, in Staphylococcus aureus, the protein binding, penicillin binding protein can also, as a result of mutation, form the PB2A, which has low affinities for for methicillin. In some cases, it could be as a result of reduced antibiotic accumulation, or which can include either purine alteration or efflux pump. And lastly, but not limited, they can also show resistance to these antibiotics by persistence biofilm formation. These bacteria, after getting this resistance, they can actually proliferate across the environment, finding a secondary reservoir where they can cause, and before you know, and this can, as you can see from the illustration, the misuse or broad use of antibiotics by humans can make most of the bacteria that's inherent in them gain this resistant mechanism, and this can be passed through their faces into the sewage, and improper sewage disposal, sometimes even proper sewage, these bacteria can be found in the environment. In the same way, it can also overuse or broad use of these antibiotics in veterinary medicine can make most of this bacteria present in some of these animals gain this antimicrobial resistance, and they are excreted in their manure, and they are disposed to the environment. Some of these bacteria too can come back, leave the environment by when they eat some of this, or the animals drink or consume some of this water that have been contaminated with these antimicrobial resistance bacteria, and the cycle continues. Today we are looking at an important river, which is Rio Soto La Marina, here at Tamaulipas. The river is located at the northeast of Mexico, and as you can see, it drains into the Gulf of Mexico. Upstream of the river, we have a lot of rivers, which include San Carlos, Pylon River, Purificación River, Corona River, San Felipe, Arroyo Grande, and San Marcos River. At the middle, we have the Vicente Guerrero Dam, which is an important river here in Tamaulipas that have been known for its irrigation and its public use. Downstream the river, we have the Soto La Marina. The river flows down into the Gulf of Mexico. Some studies have reported that there are some waste contaminants that have been leaked into this river and have put this river at risk. The antecedents or the background of our study were studies. One of the such studies, the study that gave us this inspiration, is a study in Germany where they try to evaluate the occurrence of diversity of ESKAPE bacteria and E. coli in a slaughterhouse. They used the wastewater and some of the processed water, and they detected all the 6 microorganisms, all the 6 ESKAPE bacteria. Alongside, they also detected important ARGs ranging from beta-lactamase to the methicillin-resistant Staphylococcus aureus, and also to vancomycin-resistant Enterococcus faecium. There they were able to, after the study, they said these slaughterhouses are important reservoirs for multi-drug resistant bacteria, especially those of clinical relevance. A study in Mexico in 2022 also, they tried, when they were working to determine the frequency of gram-negative bacteria in an important river, which is Lema River, they were also able to identify, they identify a lot of gram-negative bacteria, but the bacteria of interest is Klebsiella pneumonia because it's one of the ESKAPE bacteria. They detected 2. And on carrying out a whole genome sequence on one of those bacteria, they also detected important ARGs ranging from beta-lactamase, carbapenemase, aminoglycosides, chloromethylphenyl acid transferase, and tetracycline. And they also end up saying that MDR strains and many ARGs that represent contaminants of importance are present, and they can cause environmental or public health problem. In another study in South Africa, they tried to look at the abundance of viable Enterococcus faecium, Pseudomonas aeruginosa, and Klebsiella pneumonia, and they detected those 3 in surface water. After carrying out the genome sequence, they were able to find 2 important genes, and they also said that this indicates that Nocosuма pathogens can flourish or live very well in environmental settings. Lastly, a study recently in Spain, where they tried to work to study the occurrence of Pseudomonas aeruginosa in an important river there in Spain, Irigua River. The surface water, they were able to isolate about 56 Pseudomonas aeruginosa. On carrying out, though, what they were testing for was carbapenems, because carbapenems are very important because they are one of those last resort antibiotics. Only one of the bacteria show resistance to imipenem and to meropenem. But after they carry out resistance, they discovered none of those bacteria actually show resistance to these genes. But on carrying out further process on this, they were able to determine that it was actually what causes the resistance was OPR gene and also a premature codon at position 415. And they also said these bacteria actually represent a pathogenic potential because of other things they see, so like the virulence factors and the great motility they see from this bacteria. Looking at this antecedents, or looking at all this study, we could actually say it's also possible that we can detect this multi-drug resistant bacteria in Soto La Marina because of its location. And we decide to focus more on the ESKAPE bacteria because of the lack or dearth or the paucity of information on this particular bacteria around the community or environment around Mexico. The justification of this work is we all know that urban rivers are usually affected by anthropogenic activities ranging from agriculture to recreational activity like swimming in those rivers. And this could actually lead to the introduction of some bacteria such as those of the ESKAPE family. The Soto La Marina is also known to have a lot of anthropogenic activity such as farm irrigation, fishing, or animal husbandry. And this can actually lead to the increase or a reduction in the microbiota of this bacteria, thereby introducing bacteria such as the ESKAPEs. And because ESKAPE bacteria are very important in ecosomal settings, and they are usually associated to humans and animals, those reasons were the drivers for this study. The hypothesis of this work is that we believe, due to studies and other things we believe that 50% of the samples we are going to collect we are explaining the hypothesis based on the upstream, the downstream, and the dam itself. So we believe that upstream, at least before the dam, that most of the samples we isolate, 50% of them would be members of the ESKAPE family. And we hypothesize that the Enterococcus faecium will actually be, will have the prevalence, will be the most prevalent, because a lot of studies have shown that Enterococcus is actually the most prevalent in environmental, when it comes to environmental studies. And also because of animal waste contamination. At the dam itself, we believe they will have treated the dam, and as a result of treating the dam, there will be reduction in this ESKAPE bacteria. And after the dam, because not only the water leaving the dam will be contaminated, other activity around the dam can also cause contamination. So we believe that we have an uneven distribution of this ESKAPE bacteria after the dam. The general objective of this work is we want to identify and characterize some ESKAPE bacteria in Rio Soto La Marina and also that Vicente Guerrero Dam, with the aim of looking at the characterizing the resistant behavior and the genes that are associated with this ESKAPE bacteria. The specific objective of this work is we want to assess the physicochemical parameters in this river. We want to isolate and characterize the ESKAPE strains in this river. We want to identify the antibiotic resistance profiles of this bacteria. Fourth, we want to molecularly detect the antimicrobial resistant genes in this bacteria. And lastly, we want to use the results we've gotten from these four objectives to be able to assess the ecological risks of the presence of this antibiotic resistant bacteria in this water system. This is our study design. All these rivers, we see, they converge to Vicente Guerrero Dam and down to Soto Marina, down to Gulf of Mexico. And this water was collected between November 2022, and we finished the collection around June of 2023. So we collected rivers from about 31 points. And also at this point, it's also important to also say we collected water from a control, which is outside of the hydrological subregion, far from the hydrological subregion, which we take the water, which is our control. Next, we were able to carry out the physical parameters. In this study, we only studied the pH of the soft oxygen, nitrate, phosphate, chromium equivalent, because these parameters influence bacterial growth. So that was one of the reasons why we studied them. And all the analysis followed the standard method by the APHA 1998. For the pH, we use pH paper. For the other parameters, like the dissolved oxygen, we use a kit known as the ANA kit. It has a liquid substance titled term reagent A to C. And we also, for the nitrate, phosphate, and chromium, we used a powdery form of ANA of this kit. We follow all the procedure that was given to us by this kit, and after we've done that, we add them into a glass cuvette. Then we measure these chemical properties using the ANA H18 spectrophotometer. Next, we use next-phase data analysis to look at the difference between this river and compare them with respect to the sampling points. So far, we collected 31 water from this river, and again, we collected a control making 32 water in total, 32 samples in total in this study. Based on the description of the site, before the dam, most of the rivers are all close, either on the roadside or the anthropogenic activity, or they are between housing area or recreation area. But close to the end, we have one which was very close to the dam, which was a fishing area. At the dam itself, it's also a fishing area, but we have more of this recreational activity after the dam, which is more of recreation, fishing and recreational area. So the anthropogenic activity change is after the dam. At the control itself, which is San Fernando River, this river was located in the road, on the road close to the roadside. For the basic physicochemical parameters, before the dam, the dissolved oxygen pH chromium level were normal, or at least close to the permissible by the WHO. However, we noticed some difference in the phosphate level. Some of the phosphate level like that in Corona 5 and San Felipe, we have a phosphate level that was high above the permissible level by the WHO. For the nitrates, we also have a high level in some river and also in San Felipe. At the dam itself, the water was good compared to the WHO accessible value. After the dam itself, most of the river were good, with the exception of a river in Soto La Marina point 2 or 4, which are 0.3, which are a high nitrate level. On carrying out the mean values of these physicochemical parameters, we discovered that it was Olario San Felipe that was higher than the permissible value that's permissible value by WHO. On carrying out the statistical distance, if you see if these rivers differ, the only significant difference we found was in the pH, which shows significant difference. However, the dissolved oxygen, the phosphate, the nitrates, and the chromium, most of these rivers didn't really show a significant difference in those rivers. So for the pH, the river was moderately acidic, between 6 and moderately alkaline, a pH of about 8, which shows that this water is at least, the water is good according to the WHO. Also, the dissolved oxygen we see in this water was okay, which shows that this river could actually support aquatic biodiversity. And also in respect to organic pollution, this water was at least, this water or these rivers were good. However, some high level of phosphates and nitrates we see in some of these rivers are seen in some study like Guzman et al. Shows that some of these rivers are actually contaminated with phosphate and nitrate. For the isolation and characterization of this bacteria, we use Barger's manual determination of bacteriology, where we carry out a dilution, water sample, the water sample, we carry out a serial dilution of up to 10 raised to the power minus 8. After we did that, we spread plate them using differential and selective agars. Some of these agars include Chromaga orientation, Chromaga acinetobacter, Chromaga VRE, Chromaga ESBM. Also, we use McConkey, Salimanitol, and we use biosculin. Next, after incubating them for at least 18 to 24 hours, we're able to isolate this bacteria based on the size, the shape, and the color of some of these bacteria. We're able to separate them. And next, we also try to check if they were pure and if they are viable by striking them on another chromaga for another 18 to 24 hours at 35 degrees. Next, we check for if they are their gram stain. For those ones that are gram positive, they have a red color. The gram positive, they have a purple color. Those ones that are gram negative appear red. And next, we were able to, from this gram stain, we were able to select 200 of this bacteria. We selected 100 gram-positive bacteria and 100 gram-negative bacteria, and we identified them using the VTEC2 compact system. We also carried out further biochemical characterization, such as oxidase test and motility test, to be sure of the bacteria we got, to confirm the bacteria we got. All this was done in the laboratory in the Interaction Ambiente Microorganism Laboratory. Based on the mean value of the coloniform forming unit, we use the formula coloniform forming per mil is equal to number of colonies per plate, that's the dilution factor, over volume of the inoculated sample. As you can see, at the far end is our control. In our control, we have high diversities of some of these bacteria we suspected to be members of the ESKAPE species. It grows in almost, with the exception of vancomycin resistant, with the exception from Chromaga VRE. However, we did not see any growth in biosculin, or we didn't see any growth in biosculin for Enterococcus faeces. But when we compared the control with the rivers before the dam, we discovered we have rivers that are similar to the control, that have high diversities of this bacteria that we are suspecting to be the ESKAPE bacteria. However, at the dam itself, we only have the growth in chromaga orientation and mannitol salt. After the dam, only rivers that are close to the dam also show high level of occurrence or diversity of some of this bacteria we are suspecting to be member of the ESKAPE family, but it reduced down to the river that was closest to the Gulf of Mexico. And some of the growth of these bacteria are like the growth we saw on chromaga orientation and chromaga ESBA. We are above the acceptable value by WHO, which is 100 to 100 C colony forming units per 100 mil. However, it shows that these rivers actually are not suitable for drinking. And we ought to have discovered more bacteria or more microbial bioburden in this river if we have used other media which are not selective or which are not selective for this bacteria. We will have recovered more bacteria. The highest we recovered so far was on chromaga orientation in soto La Marina 0.2, which was around 5 times 10 raised to the 8 colon-form units in 100 mil. And this could be as a result of the anthropogenic activities that are taking place in that river, which is mainly fishing and recreation. That means it's very possible that a lot of activities is going on in that particular river. In the Marconki, we have the highest, which was 2.5 times 10 raised to the 5 in 100 mil, which was similar to a study in a riverine area by Taco et al. In Voga River Basin. This result was, it shows that the Marconki, that was the highest we have, which was smaller than what we have compared to what we have on chromaga orientation. This shows that it is very possible that these rivers in Lesotho might have, there is no recent fecal pollution, or there is a reduced number of lactose fermenting bacteria. For the distribution of the gram-positive bacteria, we identified 100 using the BITEC. We discovered that one of those bacteria, such as Staphylococcus saprophyticus, was the most diverse bacteria in this river. We saw it at the dam, before the dam, and after the dam. However, so we have high level of Staphylococcus in the river, and we have a low level of Enterococcus in the river. So far, the bacteria, we isolated about 3 strains of the Enterococcus species, but none of them were Enterococcus faecium or Enterococcus fecalis. And for Staphylococcus, we isolated about 75 of them, which was the highest. And this is also possible because Staphylococcus can survive in harsh conditions, so it was not surprising. However, when we compare this study with a study in Germany by Ebbe et al., they have almost the same diversity as they have high level of Staphylococcus saprophyticus too in their river. However, we have low level of Staphylococcus aureus, which we have just one. Staphylococcus epidermis, Equerum, and Clesi, we recovered just one of those bacteria. The low recovery of this Staphylococcus aureus in this river might actually be that most of other organisms have actually outcompeted this bacteria, and that's how we have just one growth in the whole of the river. However, for the gram-negative bacteria, we have a different distribution of bacteria. We isolated a lot of bacteria, and so far, bacteria of interest, we isolated about 8 Acinetobacter species, but just about 5 of these bacteria were of interest because we have just 5 Acinetobacter baumannii. For Klebsiella species, we isolated 1. However, it was not the bacteria of interest because it was Ocytica, not pneumonia A. And for Pseudomonas, we isolated a lot, about 35 strains, and just 13 of them were the bacteria we were looking for. And lastly, for the Enterobacter species, we isolated 10 Enterococcus cloacae and 1 cancerogenous, Enterococcus cancerogenous. So in total, out of the 200 bacteria we identified with the VITEC, we are able to have 30 of them were our ESKAPE bacteria, which make 15% of all the bacteria in total. 1 of them was Staphylococcus aureus, 5 was Acinetobacter baumannii, Pseudomonas aeruginosa was 13. Then we have 11 strains of the Enterobacter species. So far, carrying out the highest prevalence of this bacteria was in Pseudomonas aeruginosa, which was about 43%. We have, the next to it was Enterobacter, which was 37%, and next was Acinetobacter baumannii, which was 17%. Lastly, we have 3% of Staphylococcus aureus. Looking at the distribution on the map, as you can see, before the dam, we have high prevalence of the ESKAPE bacteria we were looking for. At the dam itself, we did not recover any ESKAPE bacteria. However, after the dam, we recovered some of this bacteria, but not as high when we compared it to before the dam. The high Pseudomonas regional also in the gram-negative bacteria was not surprising because we know that bacteria, Pseudomonas species, can actually survive in a place with harsh conditions too, and with low nutritional requirements. They can actually survive it, so we have a high level of it. When we compare this recovery with another study in Mexico, where we had almost similar percentage of this bacteria distribution. In our study, we have 35%, they have 32%. However, most of our bacteria, we were able to isolate Pseudomonas aeruginosa, but they were not able to isolate these bacteria. For the Enterobacter species, when we compare it to another study here in Mexico, 10% of our bacteria in this study was actually Enterobacter species. In another study in Basic Falls, in National Park in Mexico City, theirs was 9%. It is safe to also say that we did not recover Enterococcus faecium, even fecalis in this river, and Klebsiella pneumonia. And this could be due to that this bacteria actually have a low growth in that particular river. And also it could also be possible that we have a methodological problem, and that's why we couldn't isolate these bacterial strains in this river. For the antimicrobial resistant profiles, we compared using the McPherson region to look at the inocular density. Then we use the BITEC 2 using broth microdilution. And also we use the efficient disk method to be able to identify, to see the resistant patterns in some of the bacteria we've isolated. For the gram-positive bacteria, we use important antibiotics. And also for gram-negative, we use ranging from aminoglycoside to even polyinse, which is colistin. Also, the results, we compared this result with the CLSI 2023 to check the resistant pattern, if they are resistant, if they are susceptible, or if they are intermediate. Next, we were able to also look at the MDR, if some of these bacteria are MDRs, by using, if these bacteria were resistant to at least 3 different classes of antibiotics, then we classify them as MDR using the Margaritis et al. Formula. For the multiple antibiotic resistance, we use Gifford's et al. Formula to be able to identify if these bacteria were actually coming from places with high or originated from high-risk contamination. Those ones that are greater than 0.2 are isolated from regions that are highly contaminated, and those ones that are less or equal to 0.2 are isolated from regions that have low contamination of the antibiotics used in this study. Also, we carry out a phenotypic detection of ESBL, MBL, and AMPC producer in this ESKAPE strain. For the ESBL, we use double-dig synergy test, and if they appear like a frog-like, then it is ESBL positive. For MBL, we use the combined Dix test method, and we use those ones, we compared the size of the EPNMM and the EPNM EDTA. The one with the EPNM and EDTA, if they are greater than the EPNM alone by at least 7 millimeters, they are assumed MBL positive. Also for the AMPC, we use this, those that look have a shape like this are assumed to be AMPC positive. For the Staphylococcus, all the reagents, all the antibiotics we use appeared susceptible. They were susceptible to all the antibiotics we use in this study. For the Acinetobacter baumannii, however, we have the highest rate of resistance in cefazolin, which was 100%, cefotroxin about 80%, cefizidim about 20%, ampicillin and sulbactam about 50%. And this study was at par or something similar with a study in South Africa, also in an extra-hospital environment. However, we have intermediate resistance to colistin, to tazobactam, piperacillin, to cefepime, and also to meropenem. They show intermediate, while the rest show that, why the rest were also susceptible to these antibiotics. For Pseudomonas have a different pattern. For also, it has a similar pattern, which was the cefazolin was resistant at least 100%, cefotroxin about 100%, ampicillin sulbactam was about 46.1%. The tigecycline was resistant to at least 4 of them show resistance to tigecycline, which was around 30.76%. Meropenem and imipenem is just 1 and 2, which were 7.69% and 15%. However, we have intermediate resistance to amikacin, cefepime, and tazopine and piperacillin. And we important antibiotics like colistins and gentamicin shows no resistance. They were also susceptible to these antibiotics. But for our Enterobacter species, they show a quite different resistant pattern, because the only one we have that was similar was the cefazolin, which was 100%. However, the cefotroxin was just 9%, cefazidine was 10%, the ampicillin, sulbactam, and piperacillin, only one show resistance, which was 9%. Other important genes like the colistins, about 3 of them shows an intermediate resistance to this antibiotic, while gentamicin, amikacin, meropenem, imipenem, and ertapenem show no resistant pattern. That is, they were all susceptible. Looking at this antimicrobial test, we used important antibiotics like the beta-lactam, cephalosporin, carbapenems, aminoglycoside. We used tetracycline, fluoroquinone, and also beta-lactams and their inhibitors. As you can see, the highest we have so far was cefazolin. In all the antibiotics we use, all the strains in this study were resistant to cefazolin. For cefotroxin, it was lower, which was 62%. For cefazolin, we have 13.8%, and for cefepime, there was no resistance. For the carbapenems, the ertapenem shows the highest of about 51.7%. For imipenem, we have about 6.9%, and meropenem, other one, which was 3.4%. For the beta-lactam, the highest we have so far was ampicillin and sulbactam, which was about 31%, and we have a low rate of resistant pattern in piperacillin and tazobactam, which only one of the 29 gram-negative bacteria show resistance to it, which was 3.4%. The tetracycline, for the tigecycline we use, only 4 of them show resistance. It was shaded because tigecycline is the last resort antibiotic, and even as minute as 4 of these microbes showing resistance to it shows that this river is a bit is contaminated. For the cefazolin we saw in this result, it has also been saw in another study in Algeria, and it must have been as a result of selective pressures by contamination, which has conferred a kind of contamination to this particular river, either from human medicine or from agricultural waste. For the multi-drug resistance, all the Pseudomonas we isolated before the dam shows multi-drug resistance. And also the one we isolated in the control show resistance. However, the Pseudomonas after the dam does not show any multi-drug resistant pattern. The acetylbioenzyme we isolated from the control also show multi-drug resistance. So in total, about 36.67% of the bacteria isolated from this river shows that they are multi-drug resistant bacteria. For the multiple antibiotic resistant index, we use the formula number of antibiotics to which this bacteria is resistant to over the number of the antibiotic tested. So far, the Staphylococcus, since it doesn't show any resistance, is the least of them. However, in Acinetobacter baumannii, one of the Acinetobacter baumannii before the river shows that it was isolated from a contaminated river. However, the one after the dam, it's below 0.2, so it was isolated from region that shows no contamination. But the two we isolated from the control shows that the MI index was above 0.2, which shows that this control was severely contaminated. All the Pseudomonas were isolated were above the MI index of 0.2, which shows that all the bacteria were isolated from place that shows contamination to antibiotics. However, for the Enterobacter species, most of them shows a very low MI index, which shows they were isolated from places that were not contaminated. Only one of them shows a MI index of equals to 0.3. So the MI index, as we have seen in this Acinetobacter, was also similar to the result in a river, in Umglen River, by Govenda et al. 2020. Also, the 19.9% ESKAPE, 90.9, because about 10 of these Enterobacter shows that they were collected from regions that are not contaminated. Only one shows this resistance. So it shows that most of these rivers have not been exposed to anthropogenic activities that could cause this resistance. And this study was also similar to the study by Edem et al. 2015. After the dam, though it was not contaminated, we didn't see any multi-drug resistant bacteria after the dam. However, we saw one of those bacteria that was resistant to last resort antibiotics like the carbapenem, ranging from meropenem, imipenem, and terpenem. And the MI index also showed that it was very high, about 0.38. And this shows that this part of the river, though they might not have multi-drug resistance, but that part of the river is severely contaminated. For the Pseudomonas aeruginosa, almost all of them show resistance, about 76.92 shows resistance, showing that this study is similar to the study by Auditorio et al. And Gouffet et al. 2019, and shows that most of these bacteria are multi-drug resistant. Looking at the phenotypic disorder of ESBL, MBL, and AFCC, for the ESBL, they were all negative. Also for the MFCC, they are negative. The only place we saw MBL positive was in the only bacteria was Pseudomonas aeruginosa, and four of the Pseudomonas aeruginosa showed this resistant, that they are MBL positive. Two of them were selected after the dam at the soto Ponente, just immediately after the dam, and the rest were celebrated from Mario Grande before the dam, and Santa Ana River also before the dam. For the molecular detection, we used the normal method, and we were able to check for those that have this resistant gene, and we were able to use the R statistical analysis to explain our data. At the control itself, the control shows that the water was severely contaminated. However, we recovered low level of antimicrobial resistant gene. Also, at this point, we also included, though it was not part of my objective, but I also included some virulence gene to have a better understanding of some of this river. So at the control, though it was severely contaminated, however, we recovered low amount of resistant genes from the bacteria isolated from the control. However, before the dam, we have more of the ESKAPE bacteria, and also at this place, we have more resistant genes before the dam when compared to the control and after the dam. After the dam, we have high level of resistance too, but not as much as when we compare them to before the dam. So far, we have 41.5% of the integrons gene in this study, that A, B, and C range between about 23% to about 16.7% to 23.3%. Also, for the variant genes for colistin resistant, MCR1 and MCR3, we have about 10% of MCR1, and we have about 70.2% of MCR2. Resistant genes such as ACRB and ACRR has a resistant pattern of 27.6% and 3.5%. We also have a resistant pattern in poster A, which is a gene that should be located in gram-positive bacteria. However, we detected it in some of our gram-negative bacteria, about 2 of them, which makes about 6.6%. In conclusion, to the best of our knowledge, this is the first research that carried out the physicochemical parameters and also antimicrobial resistance together in this river of the hydrological subregion of Rio Soto La Marina. From our study, we discovered that the river was, to some extent, was free from pollution. But however, we also detected that some of the physicochemical parameters were also high. And the growth of some of the bacteria on medias, like Chromaga ESBL and Chromaga Acinetobacter, Maconchia and Salim-Antonito, also shows that this river were not that pure, and this cannot be overlooked. In this study, we recovered very few amount of ESKAPE bacteria in the whole river, and it is also possible that some of the members of this ESKAPE bacteria might not really be able to survive outside human or outside animal environment. So maybe that's why we have a low recovery. And also, they might not have the necessary adaptation to survive in rivers, and that could explain why we recovered a very low amount of these ESKAPE pathogens in this river. Also, some rivers shows that they were not polluted, but we are able to still have the highest CFU value of Chromaga or ESBL and Chromaga orientation in rivers after the dam. They show that they were pure, they were clean, but we have high level of this CFU in this selective media that shows that bacteria that grow on them are actually very pathogenic. And also, the presence in some of this river with good physicochemical parameters, especially after the dam, also show high level of, one of the river is the SLMH, where we have the growth of carbapenems, which shows that that river might be pure, but it's severely contaminated with a last resort antibiotics, which is carbapenem. So the recovery of this pathogen from this river shows that this river, that people who are immunocompromised, when they take the fish or some of this water, it might actually cause harm to them. Also, this study reveals that the pathogens resistant genes are more with bacteria before the dam, which could also show that this, as compared to other study by Auditorio, says that wastewater might have actually contaminated this water. So it's possible that this river before the dam are actually contaminated, and that's why we could recover high level of ESKAPE pathogen and also the resistant gene in this study. Also, moreover, the presence of multi-drug resistant Pseudomonas aeruginosa and Acinetobacter in this river, and also seeing variants such as the colistin variants, so like MCR1 and MCR2, these are last resort antibiotics in this river, shows that this river are highly contaminated and may represent threats to public health. The finding in this study shows that there is a need to continue to review surrounding rivers around Rio Soto Marina and check for spread of ESKAPE pathogen using other methods. At this point, I want to appreciate the entire staff and students of CBG for this great opportunity to present. Mostly, I want to appreciate, especially I want to appreciate my supervisor, who have helped me this far, all my project committee, who has assisted me so far. Also, all the students and staff of Interaction, Ambiente, Microorganismo, I really appreciate all your help throughout in this ongoing research. I really appreciate everybody. Thank you for listening.
Bueno, agradecemos al maestro y al Departamento de Interacción la presentación que nos han dado el día de hoy y damos la oportunidad a la asistencia para que externen sus preguntas o comentarios acerca del tema que nos ha presentado el maestro. Adelante.
Thank you about your presentation. I really like your presentation style that divide per sessions. So I got two questions. The first one is, what is the relation between lactose fermentation bacteria and the fecal material contamination that you found in your bacteria?
Please, can you repeat the question? I'm sorry, I didn't get the question.
Okay. What is the relation between lactose fermentation bacteria and the fecal material contamination that you found in your results?
Lactose fermenting bacteria?
Yeah.
Yeah, in this study, one of the things was the McConkey agar we used in isolating some of these bacteria, because some of these bacteria, like the E. coli and some of these ESKAPE pathogens are lactose-forming bacteria, and the best method to use to isolate them was McConkey. However, it was not as much as some of the bacteria we recovered from, say, from the chromaga orientation. So there is a direct contamination because some of these Pseudomonas, some of these ESKAPE bacteria actually goes very well on McConkey agar.
Okay.
I don't know if I answered the question. No.
And another question is, what molecular mechanism may be contributing that cephalocin resistance in your bacteria?
Sorry?
Okay, again. What molecular mechanisms may be contributing to cephalocin resistance in your bacteria?
The resistance to cephalocin?
Yeah, the molecular mechanisms.
Mostly to cephalocin is usually beta-lactam modification. It could be any of those methods. It could be efflux pump. But mostly it's usually as a result of modification to the cell wall, because cephalocin is actually a bacteria that inhibits cell wall. It usually works by inhibiting the cell wall. So any bacteria that want to escape, the ESKAPE bacteria that want to escape this mechanism must be able to modify the cell wall to prevent this bacteria from attacking it.
Okay, and the last one. I don't know if you mentioned that, but what was the reason you chose La Marina River?
Okay. We've seen a lot of studies. A lot of studies have shown that some of these bacteria are actually present in rivers. And looking at some of the anthropogenic activity that are going on in this hydrological subregion, it is very possible that we can see this ESKAPE bacteria. And that was one of the reasons we took it, because a lot of anthropogenic activity is going on in this river, and the river is one of the important rivers here in Tamaulipas. So there is a need to understand the pollution rate or the contaminated rate of this river so that we can give a public awareness to the people and to the government.
Okay, thank you.
Thanks. Okay. Thank you.
Bueno, si no hay más preguntas, vamos a dar paso a la entrega del reconocimiento que hace el Centro de Biotecnología Genómica al maestro por su participación del día de hoy. Y me permito leer: Instituto Politécnico Nacional, Centro de Biotecnología Genómica, otorgan el presente reconocimiento a TUNDE OLARINDE por su destacada participación como ponente en la plática del Laboratorio de Interacción Ambiente Microorganismo con el tema Resistencia antimicrobiana, caracterización y análisis molecular de cepas del grupo ESKAPE de la subregión hidrológica del río Soto, Marina, Tamaulipas. Ciudad Reynosa, Tamaulipas, a 25 de noviembre del 2024. Atentamente, la técnica al servicio de la patria, doctora Amanda Alejandra Oliva Hernández, directora. Muchas gracias, maestro.